Imagine waking up in a world where inherited diseases are nothing more than historical footnotes, erased from human lineage as easily as a typo corrected on a digital screen. This is not the realm of distant science fiction, but the rapidly approaching reality of our present. We stand at the precipice of an evolutionary crossroads, holding a tool capable of rewriting the very code of life. The instrument driving this monumental shift is CRISPR-Cas9, a revolutionary gene editing technology that has permanently altered the trajectory of biological science. Originally discovered as a rudimentary immune system used by bacteria to slice apart attacking viruses, this molecular machinery was repurposed by brilliant scientific minds, most notably biochemist Jennifer Doudna. They transformed a microscopic defense mechanism into a programmable scalpel, capable of finding and altering specific sequences of DNA with unprecedented precision. To understand the magnitude of this breakthrough, consider the human genome as a vast, complex instruction manual containing 3 billion biological letters. Before this innovation, DNA modification was a clumsy, inaccurate process, akin to editing a massive manuscript with a sledgehammer. Now scientists can deploy a guide molecule to locate a precise genetic typo, allowing an accompanying enzyme to snip the double helix, initiating a natural repair process that rewrites the sequence. The initial applications of this molecular scalpel have focused on somatic editing, targeting the non-reproductive cells of a living patient to treat devastating genetic conditions. In clinical trials, researchers are already extracting stem cells from patients with sickle cell anemia, correcting the faulty genetic instructions in a laboratory, and infusing the repaired cells back into the body. This examines the transformative potential of the science at its most profound, offering functional cures where only palliative care previously existed. The ripple effects of these early clinical triumphs are reshaping the pharmaceutical landscape and offering unprecedented hope to millions suffering from rare genetic disorders. Yet, the triumph of somatic therapies carries a built-in biological safeguard, as any genetic changes made to a patient's blood or muscle cells die with that individual. The alterations cannot be passed down to their children, securely containing the biological impact within a single human lifespan. But the ambitions of genetic science rarely remain confined to treating the living, inevitably pushing toward a far more controversial frontier. The same molecular scissors that can cure a blood disease in an adult can also be applied to a human embryo, a sperm, or an egg. This practice, known as germline editing, crosses a fundamental biological threshold by making alterations that will be inherited by every subsequent generation. The moment we transition from somatic to heritable modifications, the moral architecture of modern medicine begins to fracture. Editing the human germline represents an irreversible intervention into the human gene pool, effectively hijacking the slow, random process of Darwinian evolution and replacing it with deliberate human-directed design. We are forced to ask if we possess the wisdom to permanently alter the genetic legacy of individuals who cannot consent to the modifications made before their birth. Beyond the philosophical objections, the sheer biological complexity of the human genome presents staggering risks. DNA is not a simple linear code where one gene dictates exactly one trait, but rather a hyper-connected network where a single genetic sequence can influence multiple, seemingly unrelated bodily functions. A precise edit intended to eliminate a hereditary heart condition could inadvertently trigger a heightened susceptibility to autoimmune diseases or neurological disorders decades later. These unintended biological consequences, often referred to as off-target effects, could echo through family trees for centuries before fully manifesting. If a deleterious mutation is introduced into the human germline, recalling the genetic error is virtually impossible, as the altered code will have already woven itself into the fabric of future populations. The laboratory miscalculations of today could become the inherited genetic burdens of tomorrow, fundamentally destabilizing human health on a generational scale. As the technology matures and the safety profile inevitably improves, the ethical quagmire deepens, shifting from the eradication of severe diseases to the allure of genetic enhancement. The boundary between a therapeutic cure and an elective biological upgrade is notoriously porous, paving the way for the deeply controversial concept of designer babies. Parents might soon face the agonizing choice of whether to pre-select traits for their unborn children ranging from enhanced muscular density to optimized cognitive function. This drive toward genetic optimization threatens to radically exacerbate existing social inequalities, creating a world where biological advantages are available only to those who can afford them. If the wealthy can purchase genetic enhancements for their offspring, we risk stratifying humanity into biological castes, where the unedited masses are left to compete against a genetically superior elite. The socioeconomic divides of the present would become permanently encoded into our biology. The specter of a genetically stratified society resurrects the darkest chapters of human history, casting a long, uncomfortable shadow over modern genetic research. The pursuit of biological perfection inevitably echoes the historical horrors of eugenics, where state-sponsored programs sought to engineer a superior populace through forced sterilization and selective breeding. While modern gene editing is driven by individual parental choice rather than state coercion, the ultimate destination of an artificially engineered human hierarchy remains disturbingly similar. Imagine a near-future scenario where elite universities or multinational corporations begin requesting genetic profiles as part of their admission or hiring processes. An individual's potential could be judged not by their character or accumulated experience, but by the optimized sequence of their inherited genetic code. This biological determinism would strip away the fundamental human belief in self-actualization, replacing the concept of equal opportunity with a rigid genetic destiny pre-written in a fertility clinic. Furthermore, the definition of what constitutes a genetic defect is inherently subjective and deeply influenced by prevailing cultural norms. If society gains the power to edit out traits deemed undesirable, we risk homogenizing the human experience and eliminating the beautiful, chaotic diversity that has fueled our resilience as a species. Conditions currently classified as disabilities might hold hidden evolutionary advantages or contribute to unique perspectives that drive human creativity and innovation. Managing this profound power requires unprecedented global cooperation, yet the international scientific community remains fragmented regarding the acceptable limits of genetic modification. While many nations have enacted strict bans on heritable human genome editing, the decentralized nature of biological research makes enforcing a global moratorium nearly impossible. The equipment required to execute these complex edits is becoming increasingly affordable and accessible, raising the terrifying prospect of rogue actors operating outside established ethical frameworks. We have already witnessed the chaotic reality of unregulated experimentation when a rogue scientist shocked the world by announcing the birth of the first genetically edited infants. This clandestine experiment, ostensibly aimed at conferring resistance to the HIV virus, bypassed institutional review boards and ignored the intense global consensus against heritable modifications. The incident shattered the illusion that the scientific community could effectively self-regulate, proving that the technological capability had vastly outpaced our ethical preparedness. The fallout from such rogue experiments extends far beyond the immediate health of the altered individuals, threatening to trigger a global genetic arms race. If one nation decides to aggressively pursue cognitive or physical enhancements for its citizens or military personnel, rival nations may feel compelled to abandon their own ethical constraints to remain competitive. A geopolitical struggle fought not with traditional weapons, but with optimized human biology, could destabilize the global order in entirely unpredictable ways. As we navigate this perilous landscape, we are forced to confront profound philosophical questions regarding the very nature of human identity. If we begin systematically editing out the vulnerabilities, diseases, and imperfections that have historically defined the human condition, do we risk losing the empathy and resilience born from shared suffering? The struggle against biological adversity has shaped our art, our culture, and our collective spirit, leaving us to wonder what remains when the struggle is genetically erased. The ethical tightrope we walk is suspended between the moral imperative to alleviate immense human suffering and the existential danger of playing architect with our own species. Denying a family the ability to erase a devastating hereditary disease from their lineage seems cruel and medically regressive. Yet, granting that same power opens a terrifying door to enhancement, inequality, and irreversible alterations to the human germline that future generations will be forced to inherit without consent. Finding a path forward requires a democratization of the scientific discourse, moving the conversation out of elite laboratories and into the public square. The decisions surrounding human genetic modification cannot be left solely to geneticists, bioethicists, and policymakers. It demands the active engagement of diverse voices, including patient advocacy groups, sociologists, and citizens who will ultimately bear the societal consequences of a genetically altered future. To build a robust framework for the future, researchers are aggressively developing new iterations of the technology that offer greater precision and control. Innovations like base editing and prime editing function, more like molecular pencils than scissors, allowing scientists to rewrite individual genetic letters without severing the double helix. While these advancements promise to reduce the risk of catastrophic off-target mutations, they simultaneously make the prospect of widespread genetic modification more appealing and accessible, as the precision of these tools sharpens the timeline for making critical ethical decisions rapidly compresses. We are moving from a period of theoretical debate into an era of imminent practical application, where the hypothetical scenarios of yesterday become the regulatory nightmares of today. The window to establish global norms and enforceable treaties is closing, demanding proactive governance before the technology becomes so entrenched that its momentum cannot be reversed. Ultimately, the story of this molecular revolution is not just about the mechanics of biology, but about humanity's relentless drive to transcend its own limitations. We have spent millennia manipulating our environment to suit our needs, and we are now turning that same transformative power inward, targeting the very source code of our existence. This represents the ultimate inflection point in human evolution, a moment where the created becomes the creator. The responsibility that accompanies this power is staggering, requiring a level of foresight and collective wisdom that our species has rarely demonstrated. We are holding the pen that will write the future chapters of human biology, and every edit we make carries the weight of eternity. The choices we make in the coming decades will determine whether this technology becomes the greatest medical triumph in human history or the catalyst for our own biological fragmentation. The quiet hum of the laboratory has given way to a roaring global debate that will define the trajectory of life on Earth. As we stare into the vast, unwritten potential of our own genetic code, we must balance our boundless scientific curiosity with a deep, unwavering respect for the fragility of the human ecosystem. The future of our species is no longer left to the slow, random hands of fate, but rests entirely within our grasp.